Welcome to the Deep Dive, where we take complex source material and distill it into the insights you need. Today, we are going right to the heart of the matter, literally. We're diving deep into cardiomyopathy.
We're talking about hypertrophic cardiomyopathy, or HCM.
It's an absolutely critical topic.
Yeah.
We've synthesized a lot of sources today covering the fundamental mechanisms, the genetics behind it, and the clinical tools you actually need to spot it.
Right.
Our mission is to take HCM, which is a genetic heart condition that affects about 1 in 500 people worldwide, and really explain why it causes the heart muscle to thicken abnormally, particularly that wall between the two main pumping chambers.
And that abnormal thickening, that asymmetric hypertrophy, is the signature of the disease. But the hook, I think, is just visceral. It's that terrifying event that often brings HCM into the public eye.
Yeah, you're talking about the young athlete.
Exactly. The sudden, totally unexpected collapse of a young, seemingly healthy athlete during a moment of high intensity.
That image, really, it defines the urgency of HCM. That collapse is often sudden cardiac death, or SCD. And for the vast majority of young, healthy individuals who experience that kind of tragedy, HCM is the primary driver. It has to be at the top of your list.
So if you ever encounter that scenario, a young, fit person collapses during exertion, you have to think of this diagnosis.
Immediately.
Okay, let's unpack this. If this is striking down young people who don't have high blood pressure, no lifestyle issues, then the problem must be, built into their DNA. It has to be fundamental.
Precisely. HCM is fundamentally a disease of the heart's engine, the cardiac sarcomere.
The sarcomere, so the tiny repeating unit of muscle fiber that's actually responsible for contraction.
It's the physical machine that generates force. So we're talking about a flaw in the actual contractile machinery itself.
So what about inheritance? Because if it's genetic, that completely changes the scope of who's at risk.
It does. For about 60 to 70% of cases, it's familial. It's passed down. And it follows what's called an autosomal dominant inheritance pattern.
Which, in simple terms, means if one parent has the gene,
there's a 50% chance they'll pass it to each child. 50%.
Wow. That's profound. That means a diagnosis in one person mandates immediate, thorough screening for every single first-degree relative. Parents, siblings, children. Doesn't matter how healthy they seem.
That is the single most important takeaway for screening.
Yeah.
Yes. Now, if we look at the specific genes where these flaws usually are, they're in the key proteins that regulate that contraction.
Okay, so which are the super high-yield mutations we need to know?
There are two main culprits. First, you have the beta-myosin heavy chain, or MYH7. You should think of this one as the more aggressive version.
Aggressive how?
Mutations here often lead to an earlier onset of the disease, more severe hypertrophy, and critically, the highest risk of sudden cardiac death.
Okay, so MYH7 is early, severe, high risk, the one you worry about most.
Correct. The second major player is myosin-binding protein C, or MYBPC3. This one, it tends to be associated with a later onset.
So these are the patients who might be diagnosed incidentally.
Exactly. We often see them diagnosed in their 40s or 50s. They usually present with a somewhat milder pattern of the hypertrophy.
That distinction is really key. But what does the flaw itself, this gain-of-function mutation, what does it actually do to the sarcomere? It's not just telling the cell to get bigger, is it?
No, it's telling the cell to go into overdrive. It creates a state of hypercontractility.
Okay.
Imagine you have a finely tuned sports car, but the accelerator cable is like a little bit seized, so it keeps the engine running harder and faster than it should, even when you're just idling.
That's a great analogy. It's constantly pushing the limit.
Exactly. The heart is hypercontractile. It's constantly working too hard. This drastically increases the energy required by the muscle cells, the myocytes, and they become starved, stressed, and injured over time.
And that chronic injury leads to
two pathological results: a chaotic cell structure, which we'll get to in a moment, and the buildup of scar tissue, interstitial fibrosis.
So the end product of this genetic flaw is a heart that, paradoxically, looks really powerful, but it's fundamentally dysfunctional.
We summarize it as the small, stiff, hypercontractile heart. It pumps forcefully, sure, but because of all that thickening and the stiffness from the fibrosis, it can't relax properly.
So it can't fill with blood efficiently.
Right. It can't fill during diastole, which is what ultimately leads to the heart failure symptoms. The stiffness is just as deadly as the thickening.
That sets up our central biological puzzle. If every single cell in the heart carries this genetic mutation, why is the hypertrophy, the wall thickening, so stunningly asymmetric?
Right.
Why does the interventricular septum, that wall separating the ventricles, get so massive, like 15 millimeters or more, while other walls are relatively spared?
This is a classic point of distinction. The mutation is global, it's in every cell. But the physical manifestation, the phenotype, is intensely regional.
Which tells us the mutation is necessary, but it's not sufficient. There have to be local factors that are amplifying the genetic flaw.
Exactly. We've identified 3 cooperating mechanisms, and they all center on stress. First, and most important, is that the septum experiences the highest mechanical load.
So during every heartbeat.
During every single systolic beat, the septum bears the brunt of the pressure and also the shear forces from the blood being ejected out of the left ventricular outflow tract.
So the highest workload region gets the biggest, most damaging hypertrophy. The harder you stress a compromised cell, the more it grows pathologically. Makes sense.
Absolutely. The second reason ties back to genetics, regional gene expression. Studies have shown that even though the gene is everywhere, the septum has the highest regional expression of these key mutated proteins, especially beta-myosin heavy chain.
So the highest concentration of the flawed machinery is right where the stress is the greatest. That's a deadly combination. What's the third factor?
It's the blood supply.
Yeah.
You need a robust blood supply to sustain that kind of massive pathological growth. The interventricular septum is uniquely supplied by 2 major coronary arteries, the left anterior descending and the posterior descending artery. This dual perfusion is robust enough to feed that enormous thickening.
So other regions might have the stress, but they can't sustain the overgrowth because the blood supply isn't as good.
It isn't as redundant, yeah. It takes that perfect storm of mechanics, genetics, and perfusion to create the classic asymmetric shape.
Fascinating. Okay, let's zoom in on the ultimate cause of risk here. The cellular chaos you see under the microscope, what does that septal muscle actually look like?
If you take a biopsy of an HCM heart, the pathognomonic feature, the one thing that just screams HCM, is myocyte disarray.
Okay, what does that mean?
Instead of the heart cells being neatly aligned, like parallel stacks of cordwood, they are inserted at these bizarre, chaotic angles. Myofibrils are twisting, turning, inserting perpendicular to where they should be.
It sounds like a stack of Jenga blocks that's about to collapse, not a load-bearing wall.
That's a perfect way to visualize it. And this chaotic architecture, combined with those scattered patches of scar tissue, the patchy fibrosis, is incredibly dangerous electrically.
Because these areas of disarray and fibrosis act like electrical dead zones or short circuits.
Exactly. And here's where the structure connects directly to that sudden death risk. These short circuits allow the electrical signal to get trapped in these reentry loops.
Which is what causes ventricular tachycardia, VT, or ventricular fibrillation, VF, the lethal arrhythmias.
That's it. Clinically, we can identify these scar zones using a cardiac MRI with late gadolinium enhancement, or LGE. The presence and the extent of that LGE is a huge predictor of that sudden death risk. The chaos in the scar are the primary substrate for those arrhythmias.
Okay, let's pivot to the clinical floor. This asymmetrical thickening creates this bizarre geometry, and that often leads to a really unique physical phenomenon, a dynamic obstruction of blood flow.
Right.
This is where the disease shifts from just HCM to hypertrophic obstructive cardiomyopathy, or HOCM.
Yeah, HOCM is when that obstruction causes a pressure gradient of 30 millimillis G or more. Understanding the mechanism is paramount. It's like a 3-act play driven by fluid dynamics.
Okay, lay it out for us.
Act 1. The asymmetric septal hypertrophy physically narrows the left ventricular outflow tract, the LVOT, that's the exit path for the blood.
And Act 2 is the physics lesson, the Venturi effect.
Exactly. When blood is forced through that narrow passage, it has to speed up, it accelerates. And according to the Venturi effect, that rapid flow creates a localized drop in pressure. Think of putting your finger over a garden hose. The water speeds up and creates a little bit of suction.
And Act 3, that sudden drop in pressure sucks in the nearest movable object,
which is the anterior leaflet of the mitral valve. And this leaflet gets physically pulled forward during systole. It's a phenomenon we call systolic anterior motion, or SAM.
So the valve leaflet actually hits the thickened septum.
It does. It creates this temporary dynamic block right where the blood is supposed to get out. And that block is what generates the hallmark harsh murmur.
A harsh crescendo-decrescendo systolic murmur. But the key diagnostic insight here is that the obstruction, and therefore how loud the murmur is totally dependent on how much blood is in the ventricle. It's highly dynamic.
This is the critical clinical clue. This is how you differentiate HCM from other murmurs, like simple aortic stenosis. The murmur gets worse, it gets louder, when you decrease the left ventricular volume, or preload.
Okay, walk us through those maneuvers. Why does reducing the volume make the obstruction worse?
Well, if you ask the patient to do a Valsalva strain, or even just stand up from a squatting position, you decrease the venous return to the heart. Less blood fills the LV.
Mm-hmm.
A smaller, emptier ventricle means that mitral leaflet is physically closer to the thick septum to begin with. The Venturi effect starts earlier, the SAM is worse, and the obstruction becomes more severe, so the murmur gets loud.
It's the classic trick question on a medical exam. Maneuvers that reduce volume should make most murmurs softer, but here it makes the HCM murmur louder because the geometry gets worse.
Precisely.
Yeah.
And conversely, if you have the patient squat, or raise their legs, or do a heavy hand grip, you increase preload and afterload. This swells the ventricular cavity, it pulls the mitral leaflet further away from the septum,
Which reduces the SAM.
lessens the obstruction, and makes the murmur softer.
So beyond the murmur, are there other peculiar findings?
Yeah, because of that severe stiffness, poor diastolic filling, you'll often hear an S4 gallop. That's the atria forcibly kicking blood into that stiff ventricle.
Okay.
And in really severe obstruction, if you palpate the neck, you might feel a bifid carotid pulse, sometimes called a spike and dome pulse. It reflects blood being ejected, then suddenly cut off by the SAM obstruction.
And the ultimate red flag presentation is still that sudden symptom during exertion. Angina with clean coronaries, or worse, exertional syncope. What does syncope tell us?
Syncope is a massive red flag. It tells us that during exercise, either the obstruction became so severe that the heart couldn't pump enough blood to the brain,
Causing low cardiac output.
or that the exertion triggered a life-threatening ventricular arrhythmia because of that underlying myocyte disarray. You can never ignore syncope in these patients.
Okay, this is critical. For the learner, the most confusing task is often telling true pathological HCM from things that just make the left ventricle look thick on an ultrasound. And the biggest mimic is the athlete's heart.
This differentiation is, I'd argue, the most vital step in a clinical assessment because the stakes are just so high. One is a normal adaptation, the other is a potential killer.
Right.
Athlete's heart is physiologic.
Going right into what I think is the toughest one first, restrictive cardiomyopathy, RCM. To set the stage, let's just start with that incredible analogy from your source material.
Oh, the concrete box.
A concrete box.
It's a brilliant analogy. It really captures the central problem perfectly. The heart might look normal-sized, maybe even a bit bulky, but the walls are just rigid, inelastic.
So they can't relax.
They can't relax. It's a classic stiffness problem.
So right away, we're talking about diastolic dysfunction. The filling phase is where the failure is, even if the ejection fraction looks okay at first.
Precisely. This is HFpEF, heart failure with preserved ejection fraction, but it's driven by that wall rigidity.
And what does that mean mechanically?
It means because a ventricle can't relax to accept blood, the pressure inside, the end diastolic pressure, or EDP, it just skyrockets.
And it's with just a little bit of blood coming in?
A tiny change in volume, and the pressure goes through the roof. And that pressure backs up almost immediately into the atria.
And that leads directly to the signature imaging hallmark that your source has really hammered home.
Massive biatrial enlargement.
Right, you see it on the echo, on the chest x-ray.
It's the physical result of all that back pressure.
Yeah.
I mean, the atria are ballooning out because they're trying to shove blood into a concrete box that just will not expand.
What's actually happening at the cellular level to turn this muscle into a box? It's an infiltration problem, isn't it?
Absolutely. The stiffness comes from abnormal material-like fibrosis or misfolded proteins depositing in or around the heart muscle.
And that's where we get the different types, like amyloidosis, sarcoidosis.
And hemochromatosis, right. They all infiltrate the tissue, but they do it in slightly different ways, and those differences are critical for diagnosis.
Okay, so walk us through that. Where does the bad stuff actually go in those three main types?
Okay, so with amyloidosis, you have these misfolded proteins, and they deposit between the myocytes.
Hmm.
In the interstitium, they're basically squishing the healthy muscle.
So it's not in the cell, it's around it.
Exactly. Now, with sarcoidosis, you get these non-caseating granulomas that actually replace the muscle tissue. They create these patches of scar.
And hemochromatosis.
That's different again. With hemochromatosis, the iron gets deposited inside the myocytes themselves, causing direct oxidative injury, cell death, and then fibrosis.
That's a fantastic distinction, interstitial replacement or intracellular. And speaking of amyloid, there's that legendary diagnostic clue, the amyloid paradox.
Ah, yes. This is such a high-yield clinical pearl. On an echocardiogram, the ventricular walls look really thick, often They look hypertrophied.
The pseudohypertrophy.
Exactly. But then you look at the ECG and you see these tiny, low QRS voltages.
So the heart looks visually huge and powerful, but electrically it's quiet.
Muted, that's the perfect word, because the thickness isn't muscle, it's protein. And that protein acts like an electrical insulator, so it dampens the signal.
Wow, so that mismatched thick walls, low voltage is a huge red flag for amyloid.
It's almost pathognomonic for cardiac amyloidosis.
Okay, that's a great connection. Let's move on to the systemic clues, because these RCM subtypes often show signs outside the heart, sometimes years before the heart failure starts.
Years before. And you have to look for them. With amyloidosis, think systemic protein deposition. Things like carpal tunnel syndrome.
Carpal tunnel, really?
Yes, often bilateral. And it can show up a decade before the cardiac symptoms, especially in the ATTR type. You also look for things like an enlarged tongue macroglossia or easy bruising around the eyes.
Periorbital purpura.
Right. And if you suspect it, you need a biopsy.
The Congo red stain.
And under polarized light, you see that classic apple green birefringence.
Yeah.
That confirms it's amyloid.
Okay, what about sarcoidosis? That tends to hit a younger population, right?
It does. And sarcoidosis has this weird affinity for the heart's electrical system. The granulomas love to infiltrate the conduction system.
So the classic story would be
A young adult, often of African descent, who comes in not just with heart failure, but with, crucially, unexplained AV block, heart block, plus other signs, in the lungs or skin.
Got it. And then there's hemochromatosis, the iron overload. That has a very memorable triad.
Oh yeah, this one's short and sweet. You just have to remember. Bronze diabetes plus CHF.
Bronze skin, diabetes from pancreatic damage, and cardiomyopathy.
All from iron deposition inside the cells, causing them to die off.
Okay, let's touch on those two other types, the eosinophil-driven ones, Leffler endocarditis and endomyocardial fibrosis, or EMF.
Right. We group them because they both cause this severe fibrosis of the endocardium, the inner lining, often at the apex of the ventricle.
With Leffler, the key is in the name, sort of. It's the eosinophils.
You must have a history of profound, persistent eosinophilia. Those cells release toxic proteins that just destroy the endocardium and cause scarring.
And EMF, that's more geographically specific.
Yeah, you see EMF in young patients, even children, in tropical regions like parts of Africa. And the key finding on the echo is apical obliteration.
What does that mean, obliteration?
It's like the tip of the ventricle, which should be this expandable space, is just filled in with cement. It's cemented shut with fibrous tissue, so it can't fill properly.
Wow. And I imagine with both of those, with all that scarring on the inside of the heart, blood clots must be a huge risk.
A massive risk. You get mural thrombi forming on that damaged surface, and these patients are at an extremely high risk for stroke and other embolic events.
Okay, so we've really established RCM as this high-pressure problem from extreme stiffness. Let's really visualize this now with the pressure volume loop diagram.
The PV loop. It's the mechanical signature of the heart. If you can read the loop, you understand the disease.
So for RCM, where stiffness is everything, how does the loop change?
The whole diastolic portion of the loop, the bottom line, shifts dramatically, UP and to the left.
Okay, up and to the left. Unpack that for us. What does that actually mean for the patient?
It means the loop becomes narrow, tall, high pressure, and low volume. Because compliance is so low, you can only add a tiny bit of volume. That's a low-end diastolic volume. But the pressure inside the chamber just spikes. It goes way up.
So they can't handle any extra fluid.
Not at all. Any volume shift. And that high pressure is immediately transmitted backward into the lungs, causing really severe symptoms. They drown in their own fluid.
That makes so much sense. Okay, now let's flip the diagram entirely and look at the opposite problem. Dilated cardiomyopathy, DCM. The weak pump.
Right. DCM is the big, floppy rubber balloon. It's defined by chamber dilation and, crucially, impaired systolic function. This is true HFrEF, low ejection fraction.
And the PV loop for this weak, floppy heart?
It shifts way out to the right, and it becomes short and wide.
So high volumes, but low pressure.
Exactly. You have a huge end diastolic volume because the walls are stretched out, but also a huge end systolic volume because the weak contraction leaves a ton of blood behind. The peak pressure it can generate is low, and so is the EF.
It's a perfect mechanical opposite. RCM is small volume, high pressure. DCM is huge volume, low pressure.
Precisely.